Okay, class. So in today's botany lecture, as we already studied in last lecture about the conditions of the plant taxonomy, now we are going to see here. Apart from little classification, we are going to our main focus will be on objectives and characteristic of the taxonomy. So first, apart from plant taxonomy, if we talk about just taxonomy, so what is taxonomy? So this is the science that is dealing with classification. Okay, so this is the first thing you have to be remember that whenever someone is talking to you about the taxonomy, the very first thing that is something that is related to the classification. Classifying something is a science which we can deal when we heard the word the taxonomy. So in biology or in any other field, systematic classification is necessary to bring an order in the apparent chaos. Actually, there are hundreds, thousands, and lakhs of the species just roaring around us in the world. And if we do not classify them, if we don't classify them, we are never going to able, we are never ever going to be able to identify them. So that's why we need this very interesting thing, or you can say the classification. Classification is very important. If you do anything in the world of biology or any other subject, there the first thing what you study that what is this thing and how we can classify. These are the two things that come in a very primitively in order to study something. So that the different type in a heterogeneous assemblies can be arranged into group having common properties. So this whole classification pattern in every aspect, okay, in every aspect like the characterization or the assessment, we what we do, we just going to place the similar kind of the characteristic in the same group and differences on the basis of differences we are going to separate them. So this whole separation and assembling of the thing that is very important inside the classification. So that's why this make classification important, because on the basis of the taxonomy and classification, we can easily see a group with having the similar qualities. And if we are not going to see something inside that group, then we will be easily going to be understandable that that thing do not have those characteristics which going to make it inside those group. So in biology, taxonomy aimed at grouping organism on the basis of mutual similarities into unit called taxa. So basically, this taxonomy thing which we see in context with the biology. So based on this, we have to see the mutual similarities. Mutual similarities like their morphology, in the sense of their anatomy, their biochemical analysis. So anything which is similar, we can put them into the similar taxa. If we talk about a singular form of the taxa, that is has to be called as taxon. But if we are collectively calling them, we can call them taxon. So the taxonomic unit or the taxon may have different level depending on the extent of the similarities among the organism included in it. So basically, all the taxas they are having their own perspective about the similarities. Sometimes some feature inside even in a group they do not match, but the degree of the resemblance should be higher in comparison to the degree of differences. Like if we take it as 100%, so in 100% scenario, similarities always has to be greater than 60%, as I think. But the scientists say that if they are having the similarity degree over the 50%, they can be matched inside a group. But sometimes the differences are not make that much a difference. So that's why we cannot put them into a different group. We just put them into the same group by having some very important similarities in between the species. So each level or rank. They also have rank, not in the only in the army they are having ranks, but our taxonomical units we are also having the ranks like the order, class, family, genus, and finally the one individual that cannot be compared less, that is species. So it has a different designation and these ranks from a hierarchical arrangement. So all these designations they come with a responsibility. So when we are talking about uh, designation is about the kingdom, they have a huge responsibility like a general in that way. And if we are talking about some order or class, they have a less kind of responsibility, but still they have some potential to hold on the species and genes. So that's why all the thing which placed inside this taxonomical units, they have their own role to play and all type of similarity basis which they can hold some of the species inside them. Now, the very important thing, objectives. Everything have objective, every study, every topic, every subject, they need objectives. Like why we are studying it, what's the main purpose to study this, why, how they will be going to applicable and what kind of field. So these are the things which should be in mind when you are going to study something. The very important thing of the objective of taxonomy we are going to see here. So basically three objectives in the case of taxonomy. First objective is taxonomy aims at classifying organism into taxa on the basis of similarity in the phenotypic characteristics where are expressed in an organism and can be examined visually or can be tested by other. So basically, the first goal is that we have to place all the species in a group and placing these species in a group only basis of their similarity. So this kind of arrangement that is really actually is very important because like there are lakhs of species, we are not going after them each and every one to see their characters. Even in place of that, we just going to grouping them and see the what are their overview characteristics. So by knowing their characteristic feature of a particular group, we are going to able all the knowledge about each and every specific species that present in it. So that's why this whole classifying inside a particular organism thing that is very important and it should be there. Now, the second thing is, our second objective of taxonomy is to assign each taxa a name. That is important. Everything, even when a baby come in the world, they need a name, and that is the first thing which come into the world. So obviously, we need name. How we will identify them? How we will calling them? How we will make them other understand what we are talking about. So that's why this naming process that is very important. So as the very first thing, what we have to do, the first thing we going to place them in a particular taxon, and second thing what we are doing about it is here. Sorry for the students. Okay, where were we? It's the naming. That is actually very important in the whole taxonomic. So the naming of a taxon, when we name something in the taxa, that is called as nomenclature. So this nomenclature portion that is very important, and this is the second major objective inside the taxonomy. Assigning a name to an organism is necessary for identifying it without confusion throughout the scientific world. So, we have to give a name for particularly for their identification. That is our primary goal. But also, why we need a name? Like some scientists in China made a discovery about a particular species. Also in India, Russia, Japan, every other country, they are also doing some same research on the same plant. But actually, they are calling it by a different name. So at that time, we are going to need a common name for all the species which are present on this living earth. So basically, this giving a name and giving a scientific name to the worldwide made this confusion clear because this stop us to do the overlap of research like one person already did the research on this, but the second person, that is from another country, he didn't know about that, because he didn't know that this particular name is specified to this species in that country. So that's why giving them the scientific name, giving them the name that is universally accepted, that should be also a primary objective of the taxonomy. So, scientific naming of bacteria, that is governed by the International Code of Nomenclature of Backs, the revised code of 1975. An updated edition and revised code has been published in 1992. So basically, this all the thing which they are telling you, that the major goal behind this, that naming should be done and should be internationally done or by a code. So the code which we have been accepted throughout many decades, this is a code of International Code of Nomenclature. In short, we call them the ICBN. So this, if we are talking about in the botanical perspective, so we can call them the ICBN, that is International Code for Binomial Nomenclature. Okay, that or botanical nomenclature, something. Binomial nomenclature, if we are talking about, then we will see it later, like how binomial and trinomial nomenclature work out. That is later we will going to discuss it. So, the third objective of the taxonomy is to serve as an instrument for identification of organism. A newly isolated organism can be assorted to its nearest ally or can be identified as a new hitherto unknown taxon. Okay, so some scientists from the world, he identifies some new species. So when he identified that particular new species, if we already had a group related to that species, so this will going to be easy when he and she analyze all the factor about that plant, then he will having or she will be having all the information about that plant. And after having gathered all the information related to the plant, we can place it inside a taxa. If we have a taxa already, okay, if we do not have classification, how we can place it somewhere? We cannot place it anywhere, right? This is going to be a specific unit or individual unit. So that's why placing all these groups, this is also important for the future studies. So the future will, in future, that another species we are going to find out, there should be a home to come. So that's why this third objective is to make a particular place for filling when we got something from the future. And sometime if they do not line with something, or they do not have a characteristic similar to other, then we put them into a different group. We can make a new example. So this whole classification thing, that should be very, liberal. We cannot be very rigid, like no new things can be come inside it. No, that cannot be done. Even our constitution is not rigid. So that's why these things has to be liable, and anything which you ever wanted to fit inside, we have a room for that. So that's why this taxonomy third objective is very clear about it. Now, this makes taxonomy a dynamic branch of biology. That's why we are calling it a dynamic branch of biology, because it has room for everyone. They are giving the primary goal for the biologists, botanists, zoologists, microbiologists, every person of the science. They have to go through with the very first step, that is classification or the taxonomy. So, because discovery of new organism constantly demanding change in the existing classification, that is also important. Like as the rules in our society, they get old with the time. So though our classification system, as the new things come in consideration, at that time we have to think about changing it. Okay, we have to adapt new things, and that is the rule of nature to live, because only those things can be lived forever, or not live forever, means they can be sustained that life after adapting something. So that's why this adaptation quality is very important. And there should be also this adaptation changing should be done throughout our classification pattern. So that's why taxonomy is a dynamic branch of biology. Now, that was the objective why we are doing classification. Now, these are some characteristics. Like everything have their own characteristic. Like plant have their own characteristic, animal have their own characteristics. So every species, everything have their own classical, not classical, they have their characteristics. So these taxonomic value, they also have their characteristic. The very first important characteristic here, the classical taxonomy. As a classical music, the same, not very different from the classical music as a classical taxonomy because it's based on some, you can say it's based on some ancient pattern with evolution pattern. Not the rigid one, but yeah, it changes slowly. So classification of any group of organism uses selected stable characteristic which vary among the taxon. Okay, so all the, we just study that out. Like the thing has to be stable inside a taxa to make it understandable and make it that has to be when we are going to describe it should be at that time, for that time it should be result. So they are known as taxonomical characteristic. So classically the bacteria have been classified on the basis of similarity in phenotypic characteristics like their morphological features, response to the grain stain, Ramstein, and cultural characteristics, physiological biochemical properties, pathogenicity, antibiotic sensitivity, serological relationships. So these all were the parameters. Okay, so classical taxonomy, if we see, is in a one form that depend on the features which has been come in existence at the primitive stages. Mostly all our classification in a primitive era, okay, not in today's world. So all the primitive kind of taxonomy or the primitive kind of classification, actually, that is based on some morphological features. And the morphological features, like you can see, if we are talking about just a bacteria. So for the bacteria, these are the phenotypic characteristics that are just read it to you, like the gram staining. So some on the basis of the staining, we can classify the bags into the two groups, like the positive and the negative. Cultural characteristics, like some can be cultured, some cannot be, or some have a very specific kind of habitat to grow. Some can be extremophils, so that basically depend on their survival nature. Physiological and biochemical properties. Every species, they have their own biochemical aura. So basically, these bags, they also have their different kind of properties. Pathogenesis. This is a bad term, but important for your classification. Pathogens means they can cause disease. So we put them in a different class. We have to put them in a different class, otherwise maybe some can go up with some good bacteria. So that's why we needed to put them into different area. Antibiotic sensitivity. So some of the bacteria, they have a special characteristic about the antibiotic sensitivity. So this whole antibiotic sensitivity is used in the pharma industry basically, and medical microbiology. Serological relationships, these are also very important, and that depend on the ecology with the normal, like blood. When we talk about serologies or the bloody things, so the blood relationships, that has to be very important when we are classifying the classical taxonomy. So taxonomically important morphological, cultural, physiological, biochemical characters we will be going to see in a table. So, the morphological character of taxonomic web. We will take it one by one. First is the cell morphology. Okay, so on the basis of this whole cell morphology, we are having this unicellular and multicellular. We all know the species can be, or even if the bacteria can be, they can be the unicellular. If we wanted to see what kind of unicellular bacteria and how we place them, like the cocci, bacilli, vibrio, spirales, foris, clostridia, stalk, sheathid, they all are the bacteria which we considered as a unicellular. Not considered, they are unicellular. Then secondly, the multicellular, they are the mycelial and filamentous. So the mycelial and filamentous, so you can see that they are having a multiple filament in them, and the mycelia. Mycelia are basically the compartments. So if they are having compartments, means they are the multiple in the cellular structure. So on these basis of the character, we can classify them in a different groups. Cell arrangement. Now we talk about the cell arrangement, how their cell is going to be arranged, like in a single unicellular that cell has no choice to be single only. Here there will be two in one chains that can be present in a chain in a long chain, bunches that is group, packets that is a packet size is compactly arranged actually. So these can be based on the cell arrangement. Staining property, I just told you, like the gram staining, if they can accept the stain, they will be called as a gram positive. If they are not going to have all the staining things, then they will be going to the gram negative. Acid fast staining, that also can we do if they are going to accept it, they will be acid fast. If they are not, not acid fast. Flagellation. This is the thing required inside the bacteria for their whole, you can say the whole, basically the motility thing and sometimes the capturing nutrition also. So the flagella is monotracheous, lophotracheous, amphitracheous, the tritidacus, endoflagellate or non-flagellate. So these kind of the flagella can be present. Monotracheous means one, proper, amphi means combination, petri means patriarchal inheritance, endo inside, non-flagellate, not no flagella at all. So these we don't have to study here all the bacteriological characteristic, but what we have to see how taxonomy can depend on that. So that's why this whole flagella thing. Motility is a movement. So on the basis of movement, how they can be varied or how we can put them in a different group. Non-motile, no motion at all. Flagellated locomotion. Locomotion will be done by the flagella. Gliding, maybe they cannot have, they do not have flagella, but they can move by gliding. Motility due to endoflagellate, or you can say the flagella, but not outside, where it will be present inside their body. So that will be called endoflagella, but motility is present. Glycocalyx, that is a capsule that present on their surfaces for their protection. So this glycocalyx that can capsule can be present or absent, and slime layer can be present over them. Spores, they are having like specific spore with the bags. Every bag have they are having some special kind of spores inside them. So if we talk about the spores, non-sporing, they will not going to have any spore. Basically, the spore is a kind of unit for the asexual reproduction inside them. Endospores formation, exospores, conidia, myxospores. These are the spores which can be formed inside the bacteria, and on the basis of these also we can classify them because they are the reproductive unit and reproduction always matters. So that's why these reproductive units are very important and we can classify the whole bacteria on the basis of these. Sporangium. So sporangium, this is where spore going to be developed. So the sporangium can be on the basis of the shape and the location of the spore, we can classify sporangium. Cell inclusions. So cell inclusions are basically like what kind of the storage material and what is the component analysis of their cell, major component analysis. That can be poly beta hydroxybutyrate, volatile polysacs, polysaccharide, and sulfur droplets, parabrosal, chlorine crystals. So these are the things which can be present inside the cell inclusions in the bacterial cell. Now the ultrastructural features. Ultrastructural features, the features can be very see by on their surfaces. So the structural surface of the cilia, here you can see the flagella, elai, fimbria, texture of the slime. So these on the basis of these also the ultrastructural features we can classify. Bacteria are so interesting. Some cultural characteristic used in bacterial classification. So the character and the variation. These are like we talked about. First parameter was morphology. So this was whole about the morphology. Okay. Now we will going to see the cultural characteristic. Cultural characteristic means oxygen relationship. So basically, on the basis of presence of the oxygen, some bacteria even cannot present in the presence of oxygen. They cannot tolerate oxygen at all. Poor choice, but it's okay. Oxygen relationship aerobic means they can be present in the presence of oxygen. Microbic, accumulative anaerobic, oblique anaerobic. So on these basis, like how they are going to choose to live in the presence of oxygen, we can vary temperature relationship. Some can be live in a very high temperature. Some cannot even tolerate the moderate temperature. Some had to be live in a very low temperature or very icy things. So that depend, and on the basis of those we can classify them. pH relationship, optimum pH, medium, acidophilic, alkalophilic. That is like the acido below the 7, alkalo more than 7, optimum pH 7. So these things also can be affect how the bacteria will be cultured. Culture media composition and ability to grow. The next parameter is here composition of the culture medium and ability to grow. So, what kind of cultural composition can be seen here? Inorganic salt medium with an organic carbon sources, medium with combined nitrogen, medium containing complex containing compound like beef extract, yeast extract. Actually, the different kind of combination inside the media can also affect the growth of the bacterial culture. Some bacteria need more carbon source, some need more sulfur, some need more phosphorus. So, what kind of the things you used in your media to culture it, to culture the bacteria? So that also basically responds in the characteristic of the bacteria. Colony morphology and agar media. So when we evaluate the colony formation of the agar plate, that is also a measurable characteristic on which we can classify. Third one is a taxonomically useful physiological and biochemical property. So physiological and biochemical property, in which what we have to see character like nutrition type. Okay, so not all the things that are going to have a nutrition by the photo. Photo means photosynthesis. So and some of these bacteria can have the nutrition by the chemogenotrophic means chemicals. Okay, photon by the photosynthesis sometime, and chemolithotrophic or gonadotrophic. So that basically depend on the reaction by which they are getting the food, and the source can be anything. So that's why this kind of the nutrition type also play a major role in their classification. Mode of energy metabolism, how they do all the metabolic activities inside their body, like respiration. If they're possessing respiration, fermentation is also a key thing for the bacteria. Most of the bacteria is known for the process of the fermentation. So the fermentation, photosynthesis, oxidation of inorganic substrate, nitrate, sulfate, respiration. So all these mode of energy metabolism that can also used for the classification. Nitrogen source utilization, how they are going to utilize the nitrogen by making it as a useful component. So that can also be go through with the nitrogen source. Carbon source utilization, how they going to utilize carbon, like sugar, acids, alcohol, organic acid, polysaccharide, whatever they are going to make, and how they can utilize that source. Pathways of glucose dissimilation, that is also a very important task, has to be performed by the bacteria in various industries, leather industry, brew industry, textile industry. So these things, how they can dissimilate the glucose, this is also a very important part, and on the basis of these also we can classify the bacteria. Taxonomically useful physiological and biochemical properties, they are very important, like the metabolic product we just see. What product actually? These not metabolism, these are the product which they can synthesized by their metabolism. So these product can be made by these bacteria. Enzymes also, like they are also very specific to the microorganism. Not each and every kind of microorganism is going to secrete the same kind of enzymes. They have very specific about their enzyme activities. Cell wall composition. Cell wall is also made up of different type of the things like the peptidoglycan. or proteins, polysaccharide, lipids. So that also depend on what kind of formation has to take place in the cell wall composition. Cell membrane. Cell wall is different than cell membrane. Cell membrane is present at inner side, cell wall present at the outer side. So outer membrane present or absent, lipopolysaccharide. Sometimes even cell membrane do not present inside the wall. That also a major point. Lipopolysaccharides, O antigen lipid, ether linked, long fatty chains, long chain of alcohol. So, what kind of membrane is present? That also matters. Sometimes it can be made of fatty acids, sometimes it is based on alcohol, sometimes it's based on antigen lipid or polysaccharides or proteins that has to be depend on the cell. Antibiotic sensitivity. So sensitivity to the antibiotics. If in the case of pathogen bacteria, as they are going to create some disease in our body, an antibiotic going to act on that. So when they started it of war, how they are going to react with antibiotics. So this is called the antibiotic sensitivity. Now the second type of the characteristic and the taxonomy that deal with the molecular taxonomy. Classical taxonomy was all about the outer things, and this molecular taxonomy that is basically depend on the very core internal structures. Core internal structure, if you are talking about, so simply, I am talking about the genetic approach to bacterial taxonomy has undergone drastic changes since the development of molecular biology in the second half of the 20th century, after 1950. The concept that the macromolecules like proteins and nucleic acid could be used as an indicator of evolution of living organisms was suggested by Jocelyn and Pauling. Actually, Pauling and Jocelyn they gave a concept. They gave a concept that the rRNA inside our body they can be evoluting. Actually. They evoluting. Evoluting means they are keeping in change. They are changing over the time period. Same species, same RNA, but they are altering their function, or you can say they are having some changes over a time period in their functions. So, on the basis of these studies, like what kind of changes is coming over a time period in a particular species, on the basis of that, we can classify the bacteria. Some have a linear changes, some even sometimes some have a potential changes, which actually change whole identity of the bacteria or even some other living beings. So in that case of the scenario, we have to see the very important aspect, a very important parameter that are RNA and DNA as a taxonomic parameter. So they describe the macromolecule and molecular chronometers because the sequence of monomer in them have changed slowly and randomly and number of changes in the particular macromolecules has increased linearly with the geological time scale. So the same thing I just explained you, like the macromolecule composition inside a cell that has can be changed over a time period, over a geological time scale. So, for the variation inside them, for the changes they have been made from the past time, they can be even analyzed by reading them out, and on the basis of these changes, we can make make a taxonomical parameter to classify things in between them. Now, the principle of some of the methods are briefly described here, like the DNA based composition. Also, in the molecular techniques, this is also very important that we has to take in consideration about the DNA based composition. Like mostly the DNA have this two kind of the base pair, GC plus AT. Okay, so A plus T and G plus sequenin plus cytosine, adenine plus thymine. Mostly the ratio of GC is always more as comparison to the ET in a biological species. So this ratio, that the ratio between GC and AC, on the basis of this we can even analyze what kind of or what classification we can make. Like in bacteria, here you can see The ratio is conventionally expressed as GC moles percentage. Organisms which are closely related, like the strain of a given species, have close value of the GC moles percentage. In bacteria, this value varies from about 25% to 80%. So actually, the concentration of the GC molecule in a particular species, like some have the 25, like they just said, 25 to 80% of the GC content can be varied inside the bags. So it can be like 30%, 35, 40, 45, 50, up to 80. So on the basis of this composition, we can classify the bacteria. This is also important parameter for classifying nucleic acid hybridization. So this is a DNA-DNA hybridization basically. Whenever we talk about hybridization, there two things have to be make one. Okay, this is a simple form of explaining hybridization. So more reliable information about the similarity of the genome in two organisms can be obtained by DNA-DNA hybridization. Like if we have a confusion, if we wanted to think like two DNA, they are having some similar things. So just say it by experiment, by you are just thinking over it that they are same by the DNA. Just what do you do? Take one DNA from a species one, take second DNA from species two. Make them from double stranded to single stranded, denature them and see if they hybridize or not. If they are going to hybridize, they will definitely same. If they are not going to hybridize, they will not. Okay. So that is very simplest form we can classify them on the basis of their DNA analysis. So because formation of a heteroreflex between two single-stranded DNA molecule derived from two organisms depend on the degree of complementarity of the single strand. Because the whole hybridization process that basically depend on the complementation angle between these two DNA. So that's why this is very important, that complementation thing inside the nucleic acid hybridization, and if they are going to hybridize, they are definitely same. You can see some variations of GC moles percentage of bacteria, like 30 to 36% Clostridium bacillus, 38 to 40% Clostridius lactobacillus and Streptococcus spirillum, 50 to 54% is Enterobacteria, 55 to 70% is Azotobacter, Algectinus, Agrobacterium, Pseudomonas, Vibrio, Spiro, and Mycobacteria. They consist of GC bacillus of 55 to 70%. And the last, but the important, 70 to 80% in the Micrococcus and Acetinomycetes. So Acetinomycetes and 70%, they are having the highest GC mole percentage inside them. So these GC mole percentages, they can be also an important aspect of variation or the classifying something that is, and it is important, not a very random kind of parameter you think about, but it is a deep and important and intimate kind of parameter which give us a very important knowledge about a genetic makeup of a particular species. This another thing, ribosomal RNA homology. How can we forget ribosomal RNA? That is specific. That is like signature sequences, means specific to every species. So an important discovery made in 1965 revealed that all living organism DNA segment transcribing our ribosomal RNA has changed more slowly in course of evolution than the rest of the genome. In other words, the ancestors are more conserved in comparison to the gene encoding proteins. So basically, everything is changing over a time period, whatever it is. Like we just see we just seen about the rRNA sequences or the concept that was given by the polling. So this discovery made that all the thing has been in the transcription. But this rRNA, the 16S rRNA, basically, they are like the signature thing. They do not change. They are very specific, and their rate of change inside them, this is very low. So actually they are having the intact knowledge from the ancestral time to the present. So that's why this ribosomal RNA homology, that is also very important. Or this thing, this non-changing thing over the time period, this is make them an important point to be considered when we are doing the classification or when we are doing the taxonomy. So So this was all the parameters that has to be considered for the taxonomy, like the DNA hybridization or RNA, ribosomal RNA. These are different, okay? RNA, ribosomal RNA. They have at least some differences. And the molecular thing, not only the molecular parameters are important, but also the classical parameters are also important, which deal with the whole outer portion and some physiological function as well as. So these all things we have to take in the consider. Apart from everything, we really need to see all the all the parameters from every direction to before putting some species inside a group. Because if you just put a species inside a group without getting all the results and without doing all the experiment and without even seeing from all the parameters, you're surely going to make a mistake, and that will can cause possible harm in the future. So that's why we have to analyze in each and every step before putting a species inside a group. So this is a whole phenomena of the plant taxonomy, not even the plant taxonomy, it's a taxonomy actually, like how we can classify something and what are the parameters should be done or what are the parameters should be considered before analyzing something, before classifying something. And I think you got all understand by having an example of bacteria for taxonomic classification. Okay, class, that's all for today. This is some references and bibliography. You can also go through with them for if you wanted to study about more about taxonomy. Even if you have some questions, you can ask me. Thank you for today.